Hello again. Now we're on part C. This is the final part of chapter 3 on cells. And this is just going to go through the various cellular organelles, and we'll start out with the cytoplasm. This is between the plasma membrane that defines the outer boundary of the cell and the nucleus. The components will include the cytosol, the cellular organelles, and various inclusions, things that happen to be found in here. So sometimes we use cytoplasm and cytosol interchangeably, but here they're actually including all of these in what's called the cytoplasm, everything that's inside that cell. So various cytoplasmic organelles, we're just going to kind of zip through these so that you will review the various organelles and what their functions are. I like to look at the cellular organelles in relation to what we call, there's actually a cytomembrane system. And so there's membranous cytoplasmic organelles. That means that these organelles are bound with cell membrane. It's that phospholipid bilayer, just like the plasma membrane that defines the cell, that goes around the cell. So the membranous cytoplasmic organelles include the mitochondria, perioxysomes, and lysosomes, which are actually vesicles that contain various structures, or various chemicals. Lysosomes contain digestive enzymes. Peroxisomes will actually detoxify free radicals that are produced during metabolism. Then we have endoplasmic reticulum and the Golgi apparatus. All of these cytoplasmic organelles are bound with membranes. The non-membranous cytoplasmic organelles include the cytoskeleton that gives the cell its structure, centrioles, and ribosomes. So membranes allow crucial compartmentalization, and it's also going to provide what we may think of as an assembly line method for cell product via this cytomembrane system that I just mentioned. So mitochondria have a double membrane structure with the inner folded shelf-like cristae. These are called the cristae, and they provide most of the cell's ATP via what we call aerobic cellular respiration, which means it requires oxygen. Aerobic refers to the using oxygen. Mitochondria also contain DNA, RNA, and ribosomes. And this is one of the clues that led scientists to come up with what we call the endosymbiotic theory, where in ancient times a mitochondrion was similar to a bacterium. It lived on its own, and it was at one point in time it was engulfed by a larger cell, and because they mutually were benefiting from this situation, the mitochondria just stayed inside of cells, and as those cells divided and evolved into other types of multicellular organisms, the mitochondria stayed there, and they provide necessary energy for that cell. Mitochondria also divide via fission, just like bacteria. And here's the mitochondrion. You can see the inner cristae, the folded membranes, and then that outer mitochondrial membrane here. It is this along these folded membrane, the cristae, where we have our enzymes for metabolism. Remember, Krebs cycle occurs in here, and then the electron transport system, those enzymes are aligned along this membrane. Ribosomes are little granules. When you look at a photomicrograph of generally an electron microscope, you can see these little granules in the cell. These are made out of protein and ribosomal RNA. Their big job is protein synthesis. Free ribosomes actually synthesize soluble proteins that will be used within the cell. They will be floating around in the cytosol, and these can be used to make proteins for other cellular organelles. There's also membrane-bound ribosomes that are bound to the endoplasmic reticulum, and they make that endoplasmic reticulum appear to be rough, I'm using air quotes. There's little dots all over it, and these ribosomes will synthesize proteins that are going to be incorporated into the cell membranes, into lysosomes, and also those that are going to be exported from the cell. The endoplasmic reticulum is a system of interconnected tubules made of membrane. It's continuous with that outer nuclear membrane. Remember, there's a nuclear envelope where there's actually two phospholipid bilayer membranes, and this endoplasmic reticulum starts basically its journey throughout the cell, first attached to that outer nuclear membrane. There's two types: the rough endoplasmic reticulum that is studded with ribosomes, and then the smooth endoplasmic reticulum, which does not have ribosomes on it. Rough ER, again, the external surface is studded with ribosomes. It manufactures all secreted proteins, those that are going to be released from the cell. It also will assemble proteins along that endoplasmic reticulum interior. The proteins are going to be assembled, and at the very end they will be enclosed within a little vesicle that's a membrane-bound sac that's going to then travel to the Golgi apparatus. The smooth ER is actually a network of tubules continuous with the rough ER, and it has enzymes that are involved in lipid metabolism, cholesterol and steroid-based hormone synthesis, etc. They're also involved in absorption, synthesis, and transports of fats, detoxification of drugs, some pesticides, etc., converting glycogen to free glucose, and the storage and release of calcium. So they're discovering a lot of roles for the smooth ER. And here is a photomicrograph that's colorized. You can actually see the rough ER studded with ribosomes here, and this would represent the smooth ER. Here's a little diagrammatic view or a cartoon drawing, and this one is the actual electron micrograph. The Golgi apparatus. The Golgi apparatus is a stacked, flattened, membranous sac structure. So what does it do? It modifies, concentrates, and packages proteins and lipids that it receives from the rough ER. And here's a picture of the Golgi apparatus. Many vesicles that are right here, they're in the process of pinching off. First of all, from the rough ER, little vesicles carry the product to the Golgi apparatus, and then after the product is modified here, then the vesicles will pinch off. A little bit of membrane pinches off, turns into a vesicle, and will carry or ship that product to another destination. And this is an electron micrograph showing you that it actually looks like the little drawings that we typically see. This is 90,000 times magnified. And here it's showing you the sequence of events from protein synthesis on the rough ER to the final distribution of those proteins. We call this the endomembrane system. The protein containing vesicles pinch off from the rough ER, migrate to fuse with the membranes of the Golgi apparatus. The proteins are modified within the Golgi compartments, and then the proteins are packaged in new vesicle, and particularly different vesicle types, depending on their ultimate destination. And this is a cool picture of the endomembrane system. Lysosomes. Lysosomes are spherical membranous bags or sacs that contain digestive enzymes. They're safe sites for intracellular digestion. So you don't want these digestive enzymes sent all over in the cell because it could damage cellular organelles. So when something that the cell needs to get rid of is present, it can be engulfed in a lysosome and then just broken up into little pieces. These digest ingested bacteria, viruses, and toxins. They degrade non-functional organelles. They have various metabolic functions, like they can break down and release glycogen, so they break down glycogen actually to release glucose. They destroy injured or non-useful tissue, and they break down bone to release calcium. So when your blood calcium level drops, there are lysosomes in the bone tissue that can actually, and it's actually through little cells that are osteoclasts. They have these lysosomes which will break down that bone tissue so that you can get that blood calcium in your bloodstream to go back up to normal. Here's showing you an electron micrograph of lysosomes. The cytoskeleton. This is a series of rods throughout the cytosol. Proteins are going to link these rods to other cell structures. There's 3 basic types: microfilaments, intermediate filaments, and microtubules. Then we have the centrosome and centrioles. These will generate microtubules to form the mitotic spindle. If you'll remember, with mitosis, to guide the chromosomes to the metaphase plate, for example, and then during anaphase, these mitotic spindle structures will actually guide the chromosomes there and will be active in the process of mitosis. The centrioles are involved with cell division as well. And also there are centrioles that will form the basis of cilia and flagella, which are structures on the outer surface of certain cells. Here's showing you the centrioles with microtubules, and this is these are going to be involved with during cell division. One goes to each pole of the cell, and that's where the spindle fibers will come out from to go to attach to the duplicated chromosomes. And here's an electron photomicrograph. It's amazing. They've actually gotten photographs of these proteins. Cellular extensions, these include the cilia and flagella. They are whip-like motile extensions on the surfaces of certain cells. They contain microtubules and motor molecules. So these little proteins actually move back and forth. They're like little motors. The cilia move substances across cell surfaces, and flagella will propel whole cells. The only human cell that has a flagellum would be a sperm cell. And they're showing ciliary function here. There are cells lining the airways, like the trachea, that have cilia that go toward the lumen, or the space inside where the air is going. And actually there's a lot of mucus-producing cells that produce mucus. Any debris that's breathed in will be trapped by the mucus, and the cilia will beat upwards toward the pharynx so that then you can get that mucus and those foreign bodies out of your airway. Microvilli are a different type of cellular extension. These are tiny finger-like extensions of the plasma membrane, and these are found in the small intestine, and they increase the surface area for absorption. And here's a photomicrograph, an electron micrograph right here showing you these microvilli, and they greatly increase surface area. The nucleus is the largest organelle, and it contains your genetic library. This contains blueprints for nearly all your cellular proteins. The nucleus responds to signals, and it dictates the kinds and amounts of proteins to be made. And this is showing the nucleus. There's a double membrane nuclear envelope around it, and this is showing some of the rough endoplasmic reticulum that is around it. So this is, again, the nuclear envelope is a double membrane barrier that encloses the nucleoplasm, or the material inside the nucleus. The outer layer is continuous with the rough ER. There are also nuclear pore complexes that regulate the transport of large molecules such as RNA into and out of the nucleus. RNA actually just goes out of the nucleus, but other large molecules may need to get into the nucleus as well. Nucleoli. That's plural for nucleolus. This is a little dark staining spherical body within the nucleus, and it actually is active in ribosomal RNA synthesis and ribosome subunit assembly. Chromatin. These are thread-like strands of DNA, and this DNA is wrapped around histone proteins, which are like little spools. Just as you would organize thread around spools, the histone proteins are the way that we organize these threads of DNA. It also has some RNA on it too. The functional or the fundamental units are called nucleosomes. Histones pack the long DNA molecules, as I mentioned here, and they are involved in gene regulation. So only certain areas of the DNA may be available to at a time in order to reveal a certain gene region if the cell needs to use that to make a particular kind of protein. The chromatin will condense into bar-like bodies called chromosomes just prior to cell division. And this is showing chromatin structure. It's wrapped around these little histones, and then this is showing a condensed, first of all, before cell division, this will duplicate to make 2 strands of chromatin, and then it will condense up into these little fuzzy-looking X's. And that is the end of Part C. I'll see you with Chapter 4.